Ma'am, you are not audible. You are not audible, ma'am. No, madam, you are not audible. Ma'am, you try again, not audible. No, not there, sir. Sir, audible हो रहा है क्या? क्लास उधर बैठे हैं. Ma'am, still not audible. No, madam, no, not coming. Without your phone, you can start, ma'am. No, no, some problem is there, sir. Huh? Volume. Not audible, ma'am, still. You told that. You told that. So we are sorry for some network issue. So just will start within 2 minutes. So before going to start this session, we are very much delighted to invite our eminent speaker of today's scientific session one of day 4, Mr. Ramesh Adepus sir. So he was a bioanalytical scientist and involved in the research and development of different spectroscopic methods and chromatographic methods, which are mainly essential for the drug discovery process. So Sir has completed his M Pharmacy from JNTU Hyderabad, B Pharmacy from Bagdevi College of Pharmacy Warangal, and Sir is pursuing PhD and also in final stage of Viva under Mewar University, Rajasthan. So Sir is working currently in Waters Private Limited, Hyderabad. So Sir has immense experience in the field of analytical and drug development process. So with the brief biodata, I'll invite our eminent speaker of today's scientific session, Ramesh sir. Please continue, sir, with your presentation. Thank you, sir.
Thank you, sir. Thanks for the nice introduction. So I will switch off my video. It's for better bandwidth. So I'm switch offing my video. I will continue my presentation. Thanks for the nice introduction.
I'll manage, sir.
Good morning, one and all. I'm very glad to be here with you for today's session. So I will take 1 hour session. So in that, I'm going to explain the drug discovery process. Can you able to see my screen, sir?
Yes, sir, it is visible, sir. You can continue.
Okay. Once again, good morning, everyone. This is Ramesh. So already I have given my introduction. So once again, I'm giving my introduction. So now I am working as a senior product specialist for MS product special. So I'm the specialist of mass spectroscopy. I'm dealing with all technical sales to the analytical Waters India company. So I have the research experience of analytical experience. I'm having the 14 plus years experience in industry. So earlier I worked in the bioanalytical, so it's a R&D, clinical R&D. So in that I used to handle the clinical trial samples like phase 1, phase 2, phase 3 samples. So from the blood and urine or maybe either the tissue samples, we are able to identify and we are quantifying the drugs. So how much quantity drug is there in the particular blood sample. So it's coming to the complete drug development process like in the clinical stage process. So today I'm going to be discuss about, so in drug discovery process, so what are different types of instruments we are using. So particularly I'm going to be explain about the mass spectrometer. So what type of mass spectroscopies are meters are available in the market and how we choose based upon the application, how we choose type of mass spectrometer and what is the role of that mass spectrometer in the current application. So coming to the drug development and process, so everybody know that it's a very huge and lengthy process. So that strange thing is that in India we are not coming to the drug discovery process. So we are collaborating with other foreign companies and doing the drug discovery process. But in India we don't have any IND process started till date. Why? Because it's very expensive process and very lengthy process. So this slide I have taken from the US FDA side. So when one drug is coming from initial stage to the approval process, how many years it will take and what are the steps involved in that process. So you have to see the discovery and preclinical, it will take minimum 6.5 years. Then it will go into the clinical trials, human clinical trials and phase 1, phase 2 and phase 3. So after that, the collected data which are filing to the FDA, then they will process the review and they will approve the process. Then it will take minimum 1.5 years. After that, Again, it is going to the phase 4. Phase 4 means post evaluation studies. That means post marketing studies means even though it's a long term, the drug using, is there any adverse events affecting to the humans? So that is called as a post marketing studies. So next slide. So what are the different stages? So in the initial stage of drug discovery is a very critical and very important step. Why? Because first we have to identify the target identity. The target identification is the most crucial part in the drug development. Why? Because so the where the problem is coming. So that means the target identification is the disease organism. So in the disease, how it is affected the disease, or maybe either it may be bacterial infection, or either it may be viral infection, or either it may be internal gene destruction, or it may be DNA sequencing. So they have to identify what cause it is affecting, like why the target is why the organ is dysfunctioning. So we have to repair that organ. So that is called as the target identification. So in that target, they will identify maybe is the protein, or maybe sequence, protein sequence, or maybe is that cells. So after that, you have to take the lead molecule. So either it may be a synthetic process, either it may be natural process, either it may be extracted, whatever it may be. So you have to select one lead, and that lead going to be hit to the particular target. That is very crucial role. So that is the reason it is taking minimum 3 to 4 years for the development of identification of target and lead. So after the lead identification, then it is going to the lead optimization. So the initial lead identification and lead optimization, more like all the department like pharma as well as biotechnology as well as microbiology. So all the departments are collaborative work, and they will participate everything, and they will conclude. So after that, the lead optimization will occur. So once the lead optimization will occur, then it's coming to the pharma role. So that particular real lead we have to formulate. Either we have to formulate in the tablet form, or maybe in the capsule form, or maybe in the syrup. Either it may be like a suppository base, or either it may be any cream or ointment base. So that is the very challenge. So the drug in which form it is stable and which is directly hit to the target, that is very important. The target lead optimization and method development process is very like crucial role. It is taking too much time. So after that, everything it is going to the in vitro studies, outside the body. In vitro study means outside the body, like laboratory tests. They will do the dissolution tests and the solubility test, as well as the stability test. Everything they will do. Then it will go to the preclinical. So coming to the preclinical, so mainly the preclinicals is animal studies. They have to select minimum 2 to 3 species, like rabbits or mice or mouses. They will select. In that, they will inject the particular drug, whatever they develop the lead. So that molecules. So it only not 1 or 2. So the initial stage it will be 5,000 to 10,000. Then it is coming to the clinical, maybe around finally they will select only 500 compounds to the preclinical. So in the preclinical, they will test in different stages. Mainly in the preclinical, they will go for the ADME. ADME means absorption, distribution, metabolism, and excretion. So in that, they will check the safety first. So whether it is directly it's going to the target or not, even though it's going to the target, so is giving any adverse event or not, they will check everything. So after that, they will formulation, different of formulation they will check, and what is the bioavailability of that particular compound, and even the dose range, different dose ranges also they will take. Then after that, they will give the IND. IND means investigated new drug application. They will apply to the regulatory body. Either it may be FDA, either it may be ANVISA, Europe, or WHO. They will submit all data to regulatory authorities. Then they will review, and finally they will give the approval to the human clinical trials. So now it's coming to the stage human clinical trials. In human clinical trials, so here the thing is, human clinical trials, we have to select the different stages, like phase 1 clinical trials, phase 2, phase 3. The phase 1 is the healthy human volunteers, limited members of healthy human volunteers they will take, and they will give the doses to the particular persons. Then they will collect the blood samples periodically, like every 1 hour, 2 hours, depends upon their Cmax level and half-life levels. So they will collect the blood samples, and they will analyze the blood samples, and in each time point, each time hour, so what is the drug quantity in that particular compound. So then it will give the flat, like what is the Cmax level and what is the half-life of that particular compound. Then they will calculate, and even though how much levels it is going. Then phase 2. Phase 2 is again, it's like patient studies. They have to select the limited members of patients in the phase 2. Then they will inject the doses. After that, they will collect the periodically samples and they will evaluate. So after that, then phase 3. So in phase 1 only they will see. So of in healthy human volunteers, no. So that is the reason. Simply they will go. They will not see the safety. So in healthy human phase 1, they will see the only safety. But coming to the phase 2 and phase 3, they want to see the efficacy. Why? Because these are the patients. So in the patients, they have to show the efficacy of the particular compound. Then only the regulatory body will give the approval to the particular drug. So after that, after the clinical studies over, then it will go to the again FDA review. Then it's a new drug application, then AND, abbreviated new drug application, or BLA, like biological license applications. Then FDA will give approval. So even though after approval also, the drug is entering to the market, but latter part also, they will continue the studies. Why? Because the post-marketing adverse events, anything else. So here the entire thing where we have to use the analytical equipment like HPLCs or GCMS or LCMS or other equipment. Like in drug development and discovery and lead optimization process, you have to identify the lead. So in that cases, UPLC, HPLC and LCMS is important. And the optimization means complete characterization. So in the characterization means that whatever the impurities are there in the compound, that everything you need to identify. Even the impurities are there, how much quantity it would be there, you have to identify, you have to report it. So some impurities are genetically effect, so that is called as a xenotoxic impurities. You have to identify the xenotoxic impurities. In that stage, you have to use only the equipment LCMS, because you have to identify very low level of identification. Then coming to the preclinical, all preclinical samples, the blood samples, you have to extract the blood, and you have to identify by using of mass spectroscopy only. There is no other way. You have to use only the LCMS technique, and the clinical development also you have to use only LCMS technique. So why? Because the most selective technique. So that each stage of drug development, the LCMS is very much important. So coming to the mass spectrometry. So what is mass spectrometer? The simplest way, mass spectrometer is like analytical instrument. It measures the weight of the molecules. How it will weigh the weight? Whether it is kg or grams or milligrams? No. So the weighing factor is like mass to charge ratio, that m/z value. So the measure of masses of individual molecules which have been converted to the gas phase. So the ions are present in the compounds that are present. It is in the liquid phase. The liquid phase you have to evaporate. It is converting to the gas phase. In the gas phase, the ions are entering to the quadrupoles. That in the quadrupole you have to measure the ions. So who uses MS? So in the in previous slides I explained, in all pharmaceutical industries they will use the mass for the drug identification and quantification, as well as impurities identification and quantification and characterization. And coming to the food, nowadays in food also, in food and environment market also, everybody is using the food. Why? Because so now they are using as a banned pesticides and banned antibiotics. So like in chili powder and rice and even oils also, there are restricted, that banned pesticides are there. So why they are using that banned pesticides? So it's very harmful to humans. So that is the reason government had banned. Even though it's banning also, somebody has procuring from other different sources and they are using those pesticides. Why? Because those pesticides are giving very high yield. So that is the reason they are using those banned pesticides. So we have to identify those banned pesticides, and you have to reject that food material. So for that reasons, nowadays it's a mandatory, even the regulatory is also, FSSAI is strictly monitoring those impurity analysis as well as the pesticides and antibiotics in meat and meat, fish and fish products. And coming to the chemical materials, in the chemical materials also like petroleum studies and paint industries, as well as in the rock industries, so everybody they are using the mass spectrometer. And health sciences is nothing but all the universities research purpose they are using. So why we used MS? So why means the confirmation of expected products is very high, and it's a very well known accepted by the regulatory agencies. So that is the reason we are using as a LCMS technique. And identification of unknown species, and the in compound synthesis process you have to check the raw materials and ingredients, and the reaction confirmation, so whether the reaction has formed or not, if formed how much percentage it was formed, or any other byproducts they are forming. So everything they have to see by using of mass spectrometer. Then purification, then characterization work. So coming to the drug discovery and development process, that metabolism and DMPK, ADME studies and degradation studies, degradation nothing but QC stability studies and the bioavailability studies. So why advantage of mass spectroscopy coming to the normal techniques, like earlier they are using like UV or maybe other spectroscopic methods, or nowadays everybody is using either LC, like a liquid chromatography system, HPLC or UPLC, or maybe LCMS. So compared to the LC is like a chromophore. If the compound is chromophore, it will come to coming to the in LC mode. So if even though if this the compound is not chromophoric, we have not performing with the HPLC method. So then you have to go with the either GCMS or LCMS. So GC also they have the limitation with thermolabile compounds. Then everybody is coming adapted to the LCMS method. It's very easy adaptable and very selectivity method and highly sensitive. So sometimes you have to go in the bioavailability studies. So you have to quantify the drug levels in the picogram levels. Generally used to calculate in the mg, milligrams levels, or microgram or nanogram. But in some cases, in few of the drugs, you have to go the picogram levels. So picogram levels, the sensitivity only the MS only, it can go achievable to the MS mass spectrometry only. So coming to the basic schematic diagram, this is in wherever you have to see in the books and like in research papers. So simply they will give the schematic diagram how it would be. So in the mass spectrometer consists of three important portions. So one is like ion source, and second is analyzers, and third part is detector. So the sample introduction, either it may be LC or either it may be GC. GC means gas chromatograph and LC means liquid chromatograph. So the liquid sample is entered into the ion source. The ion source, it will take the ionization. So either it may be the compounds, depends upon the compound nature, whether it's a compound is polar or mid-polar or non-polar. So based upon the polarity, we have to choose the different type of ionization techniques, like ESI ionization, then APC ionization, then APP ionization. So that's mass analyzers. So in my coming slides, I will focus on these two. Why? Because these two are the main important factors to select the mass spectrometry. And the mass analyzers are different types of mass analyzers are available in the market. So which type of mass analyzer we have to choose? So based upon our application. So either we have to go the qualitative work, either we have to go to the quantitative work, either we have to do the qualitative and quantitative like characterization work. So in which type of work, which instrument you have to choose. So based on that, we have to select. So in coming slide, I will explain in deeper way. So this is the schematic diagram of complete instrument part. So here the first portion is sorry. So here this is the liquid chromatography front end, and here the ionization will take place. The yellow color part is the ionization. So after the ionization, the ions are entering towards the quadrupole. So in the cone, that another second is the yellow color portion. It is showing. Actually, I want to show the presentation on screen pointer. Okay. So here the ionization will take place. After the ionization, the ions are entering towards the quadrupole. So here the cone, this is the cone, we have to apply the voltages. So based upon the voltages and potential gradient, so the ions are entering towards the quadrupole. So this portion is complete the green color, this portion is the completely is the vacuum based. So here the ion gate first, after that is the quadrupole. So based upon the quadrupole, we have to identify either it may be single quadrupole or triple quadrupole or like time of flight technology. So I will explain the deeper way. So is the schematic diagram of instrument. So here just I want to show how it will be, where the like a temperature will apply here, the temperature will apply, gases will apply, the conversion of ions will occur here. After that, entering towards the quadrupole, the first quadrupole, then it is the collision takes place here. Collision means we have to select the parent here, the parent will be fragmented here, the fragmented ions are entering towards to the quadrupole 2 and identified, and it is going to the detector part. So I already have explained there are three type of ionization techniques that takes place in the ionization source. So the most commonly used ionization technique is the electrospray ionization. So in most of the compounds having the polarity in nature, so all the polarity to the midpolar compound easily ionizable by using of electrospray ionization. So that is the reason the mostly widely accepted electrospray ionization in all type of instruments. So if the compounds are little challenging, it's not going to be ionizable by using of electrospray ionization, then they will choose the atmospheric pressure chemical ionization. So then if the compounds are not able to ionize using of ESI and APCI, that compounds are very widely, it will come the atmospheric pressure photoionization method. So coming to the mass analyzers. So in the mass analyzers, how many types of mass analyzers are available? So one is single quadrupole and second one is triple quadrupole. In between there is no double quadrupole. Why? Because so in the triple quadrupole itself, scientifically speaking, is the two quadrupoles. But generally that everybody is the terminology they are using triple quadrupole. Why? Because in the two quadrupoles they have inserted one collision cell. So in that collision cells also somebody has called as a one quadrupole. So that is the reason it comes as the triple quadrupole. Triple quadrupole or tandem quadrupole, both are the same words, synonyms. And the another technique is time of flight. And even the time of flights, the quadrupole time of flight is another technique. And is the quadrupole time of techniques with mixture of like we are mixing of different type of techniques like IMS means ion mobility system, HDX means hydrogen deuterium exchange, and MALDI, so matrix associated laser dissociation induction. So that is a like type of technology, like imaging type of studies you have to do with MALDI-MS and ETD, electron transfer dissociation. So these are different types of ionization techniques as well as analyzers are available in the market. So depends upon your application. So it's a complete full-length characterization, very innovative molecule, and you have to identify the larger molecules like protein and peptide sequence, or maybe 150 kilodaltons of monoclonal antibodies. Such type of applications you have to do with the high-end instruments like HRMS, called as a high-resolution mass spectrometer. So with techniques of IMS and HDX. So I will show. So in coming slide, just I will give the brief idea about what is single quad and what is triple quad with schematic as well as video and graphical identification, and some data also I will show with you. So this is the single quadrupole. The single quadrupole already I have shown the schematic. So here the simple the quadrupole here is the one, the ionization source and the inlet parts and the voltages and gases, that everything is similar to the single quadrupole and triple quadrupole. The only the difference is here we have only first after ion gate we have only one quadrupole. After quadrupole, in between of detector and quadrupole only one, there is no collision cell present here. So what is the use of single quad? Single quad means the application you want like compound identification, just whether the compound is present or not, like the mass is present or not, just you have to identify. So in that case, no need to go for the high-end equipments, like so the cost also will vary. Single quad to triple quad to QTOF, the cost is huge. So no, like in the smaller industries or academic, they will not bear the high end of instruments. completely. So they will go only for the low type of instruments which the purpose will solve for that application. Like they want to do the only identification of that compound. Either it may be another different impurity source or maybe not. Whether the compound is present or not. In that case, they have to use only single quad. So here the single quad, the parent ion will, like the if the compound having the mass of 200 or 250, whether it is giving the compound is present in the sample, it is giving the 200 spectra. So the compound is present, they will report in that. So what are the disadvantages? I will explain in the coming slide. So what is the triple quad? So triple quad is the schematic diagram. Here in the single quad, we have observed only one quadrupole, but in triple quad, in between of two quadrupoles, we have introduced one collision cell. Why it is one collision cell is, for example, in single quadrupole, it is giving, you have to identify one solution having the mass of 250. You have dissolved one compound, whether the compound is present or not. So the single quad will give, okay, 250 mass is there or not. But somebody has raised one question. The 250 is coming from your source, or maybe 250 is coming from other source. That may be the interference coming from the solvent source, or any impurities coming from the raw materials used for the solution preparation. Even that the raw materials having the 250 molecular mass also, it will add on to the compound. So it is giving the false positive results. So in that case, the another technology, it's a very beautiful technology, is the triple quad technology. Here what it will, the triple quad will give the is two type of identification, like parent as well as the fragment. That means 250 it will select in the first quadrupole. The 250 molecular mass, it is entering to the collision section. The 250 molecular mass, we are applying the energy. So that energy, it is breaking to the 250 molecular mass, and the 250 is broken to the different fragment ions. So the MS2 is select the fragment ions. So that means you have to selecting the parent as well as fragment. Somebody will raise the question. So if the compound 250 fragment coming from the another interference source also, it will break and it is giving the same fragment, what we can do? So one, we have the thumb rule. So one compound having different there are the unique, like fingerprint, thumbprint, fingerprint rule, that so there is no molecule having the same type of fragmentation, even though they are saying, but in the few compounds like larger or maybe proteins and peptides, they are having the similar compound, similar structure, similar fragmentation process, even the fragments also same. So in that cases, we have to go to the higher masses, like high resolution. That's like TOF technology. So I will go for the another process of TOF technology. So here the simply I will explain the slide. So here the basic difference of one quadrupole and two quadrupoles in between of collision. So how it will in next slide. So the instrument schematic diagram of is a quadrupole. So when the qualitative analysis and quantitative analysis, the better instrument is like a single quad as well as a triple quad. So complete coming to the high-end technology as a quadrupole time of technology is nothing but here the same ionization source are same, the step wave, the initial guide step wave, and after that one quadrupole one. Here the collision cell. Here the before it going to the detector part, we have the drift tube. So this drift tube called as a TOF tube, like time of flight. So time of technology, time of flight means the ions are how much time it will take it will traveling time to reach to the detector is called as the time of flight technology. So here it is the ions are traveling more up towards the detector. So it is giving the exact elemental composition of that particular compound. So how it will give in coming slide I will explain. So this is the another type of technology in quadrupole itself. We have the use of IMS technology. Why we have to go for the IMS technology, I will explain in coming. Just I have to give the schematic diagram. So here the ion mobility system. Here the difference is the before quadrupole we are introducing one ion mobility system. The ion mobility system, why we are using ion mobility here? The compound having same molecular mass, same fragmentation process, same fragment ions, even though the fragment ions also having the exact mass. So then how we can differ? How can we identify false positive results? How can we avoid false positive results from the exact results? So in that case, we have to use the ion mobility system. Ion mobility system is giving the orthogonal identification technique. It is giving you as another like CCS, collision cross section value it will give. So it is a unique identification of particular compound. It is the identity CCS values is differ from one compound to another. So how it is that I will give the one video explanation in that you can able to see. So then it is. So after that wire, we have one more resolution. So there are, I'm showing just three schematic diagrams. Why? Because these three are the high resolution, high end mass spectrometry. So depends upon your application. So how much resolution you need, whether you low resolution or mid end resolution or high end resolution, are completely research grade instrument. So Synapse is completely research grade instrument. Here you have to combine with IMS like ion mobility system and HDX. HDX means hydrogen deuterium exchange, and MALDI. MALDI means laser matrix associated laser induced technique, and ETD is electron transfer dissociation technique. So like that, we have to different type of experiments you have to attach with the instrument. It's completely research grade instrument. Even the you have to see the detector. It's a W type drift tube, where top tube. So that means high resolution will occur here. So this is the instrument quadrupole. The quadrupole is nothing but it's just four parallel rod. So the opposite rods are positive and negative. So it will attract ions towards to the other quadrupoles. So we are giving like RC and DF and RC voltages. It is going to traveling towards to the detector. So this is the diagram. Just you have to the video representation see. So how it will multiple reaction monitoring means. So this is the ion gate. So the ion gate means the all ions are entering to the ion gate. The Q1 means you have to select only the parent. So here we have to see the all the ions. For example, one compound is 150 mass, 200 mass, 250, 300, 350, 400, 450 mass. Let's say examples of all these molecules are entering to. Just you have to identify only 300 molecular mass. So that Q1, the quadrupole will set to set the instrument that we have to select only 300 mass. So it will enter only the 300 molecular mass. It is entering towards to the quadrupole one. After that, we have to use the collision. In the collision, here the collision will take place. Ions are entering. 300 mass are enters. Then it is entering to the collision cell. Here the collision will occur. That 300 parent, here the fragmentation will takes, and it is forming to the like 300 mass, and the fragment, one fragment is 220 and another is 200, another is 150, another is 120, another is 100. So all the fragments are entering towards the Q3. Whether you have to select which fragment you want, like you want all the fragments, you have to select all the fragments to be entered into the Q3 specific to the parent, and or you have to only choose from parent, or from parent you have to choose only 250. Then 250 it is entering towards to the Q3. That 250 only entering to the Q3, then it is going to the detector part. You have to identify. This is the process of triple quad multiple reaction monitoring. So just I will show the video representation of this.
Family of advanced benchtop mass spectrometers from Waters. Design. advanced benchtop mass spectrometers from Waters, designed with engineered simplicity, making the highest quality data accessible to every scientist. Introducing the Zebo G2 QTOF. Groundbreaking quantum technology for the Synapse G2, which provides a new dimension of high resolution, exact mass, quantitative performance, fully compatible with new PLC acquisitions, and new ways of manufacturing. No analysis, just exact mass, quantitative and quantitative performance. Family of advanced benchtop mass spectrometers from Waters, designed with engineered simplicity, making the highest quality data accessible to every scientist. Introducing the Zebo G2 QTOF.
G2SI delivers dramatic enhancements in sensitivity with revolutionary step-wave ion optics technology. Ions is ample more efficiently than ever. At the same time, the innovative four-axis design actively eliminates lethal components. Together with the latest independent and acquisition and high-resolution multiple components, you can learn complete sensitivity
Thank you. Thank you for watching the videos. So now I'm continuing the presentation. So now we have seen that different type of mass spectrometry. So how we do choose suitable mass spectrometric system for the drug development? So the each stage, which stage you are there, like in the development stage, or maybe screening stage, or maybe characterization stage, or completely quantification. So in which type of application, so based upon the application, which instrument you have to choose. So these are the fundamental questions which raise while selecting the mass spectrometer. So which type of analysis, qualitative or quantitative? So what is the range of interest? So that means whether you have to go with microgram level sensitivity or nanogram level sensitivity or picogram level sensitivity, or you have to choose only like kind of only higher concentration of studies. So which mass accuracy is needed? So if mass accuracy is needed, how much mass accuracy you need, and how much sensitivity you need, like how much resolution you need, and even the sensitivity or specificity both required or only one. So based on that, your application, you have to choose the different models. So in earlier, I have explained all the different analyzers. So you have to choose the based upon application, different type of application. So here the thing is, the most common is that in approximately 1500 compounds are present the same molecular mass of 250. So in that case, you have to choose the single quad. You have to identify the mass of 250. The possibility of 1500 contaminants are present. So depends upon. So maybe not complete 1500 compounds are present in your sample. May one or two interference coming from other sources, maybe raw materials or solvents or any reference standard, whatever you are using, that impurity. So it comes. So in that cases, the single and multiple reaction, that single quad and triple quad, you have to choose a single quad. For example, in the pesticide identification, just here you have to identify. For example, you have to identify as the 134 molecular mass, but the 134 molecular mass is isoprotein also producing 134 fragment ion, as well as metazachlor also producing 134. If you have to identify the single ion monitoring mode, so 134 you are keeping, if the pesticide having the same isoprotein, so it is giving the false positive. In the both 134 it is coming and is giving the false positive. But so that is the reason you have to go with the triple quadrupole. The fragment ion is, the parent mass is 278. The 278 is fragmenting and it is giving the two fragment ions. So one is 210 as well as 134, are different type of fragment ions are giving. The 275 is Q1, it will select as the 278, and in the collision cell it will break and it is giving as a different fragments like 134, 210, 105. These three, you have to select which you want, either 134 you want or 210. So this is compared to the single quad, triple quad is the more specific and more accurate. So then coming to the difference between triple quad and tough. So why we have to select triple quad and tough? Here what is the main difference? The basic difference is all single quad and triple quad giving the nominal mass. So nominal mass means what? For example, 249. So C20H9, it is giving the molecular mass of 249. The same C19H7N plus also, it is giving 249. And same C13H19N3O2 also, it is giving 249. If the mass spectrometer is giving 249, so 249 means the user can identify which compound it is, either this, or either this. He will not identify as a accurate way, as a selective way. Why? Because single quad and triple quad is giving as a nominal mass. Nominal mass means carbon monoxide means 28, it will give. Nitrogen means 28, and C2H4 it is giving as a 28. But when coming to the exact mass, that is time of flight technology, it is giving as a C20H9. That all empirical formula. That means it will calculate as a monoisotopic mass of particular compound. That means if carbon is not 12, so 12 exactly is 12.00, but hydrogen is not exactly 1.0078. Nitrogen is not 14. If you have to calculate as a nominal mass, carbon is taking 12, hydrogen is taking 1, nitrogen is taking 14. So that is the reason it is giving nitrogen is 28, is 48. But you have to take exact mass, nitrogen is 28.0061. So here, if you have to take these three compounds, here 249.074 is one, C2029, H9 is 249.0704, C9. So here the top is giving as an exact mass. So based on the exact mass, either compound is this or this or this, you have to identify. So that is the major difference between triple quad and top. So coming to the top, how much resolution you need? So in the triple quad and single quad, it is giving as a unit mass resolution. Unit mass resolution, for example, if the compound is the 215 molecular mass, the above red color peak is the unit mass resolution. That means the compound is giving the only single peak with the resolution is around 400 to below 1000 resolution single quad. But the higher Q of high-end resolution, so you have to have the resolution is more. Resolution nothing but separation of two peaks. So here the compound having the two compounds here, but even though single and triple quad is giving single peak, but you have to analyze the same sample in the high resolution, it is giving as a two different peaks. Why? Because it will identify the exact mass with high resolution. So the same example here, you have to clearly identify that this is the single quad separation of 249 with compounds of three compounds are present in the single peak, and another is the fourth compound. So here they are identifying clearly separating. Why? Because one is 249 and one is 250. It is a unit mass, that means single mass unit mass resolution. That is the reason this one mass resolution identification it is giving in the better way in single and triple quad. But when coming to the same exact mass, we are not able to separate by using of LC. Means in the LC also it is giving as a single peak. Even single and triple quad also it is giving the single peak. So we have to come into the like high-end HRMS technology. It is giving the three different compounds are present here by calculating of the exact mass, as well as resolution is good. So it is separating the peak from the 249. Like there is no mixing together these two peaks. Here the well separated 250 and 249, and in between of peak 249 how many peaks are there, that everything. So that is the importance of high resolution. So the role of high resolution mass spectroscopy in drug discovery and development. Here in drug discovery development, we are different type of stages we have present. So like as this compounds in my sample, that is nothing but screening. After screening, quantification. Quantification is nothing but how much is, if the sample is present, if present, how much quantity it is there. That is nothing but quantification. So then after screening and quantification, what else in my sample? That means complete characterization and elucidation. After that, it is completely after the elucidation and characterization, then another orthogonal technique like application is comparison. Comparison means nowadays like a generic market, we are like increasing the generic market. Generic nothing but there is a slight changes in the drug, that like simply we can say as a abbreviated A&D application. The new drug application is new drug development, and A&D is abbreviated new drug. So that means they will not change the basic moiety of the compound, just they will not disturb the skeleton, just they will change the side portions of that compound, whether they are giving the they'll change the type of mode, whether it may be immediate release or sustained release or delayed release. So they will change the formulation, and they will release the same compound to the market. So that's called as a generic. So the exact formula and effectiveness of the compound is the same with the innovative compound, but only thing is the compared to the cost is cheaper. When the innovative molecule in market availability price is, for example, the market is 20 rupees per a tablet, the generic molecule tablet is coming just 3 to 4 rupees. Why? Because the innovative has did lot of work for the research. That means the lead optimization, lead identification, and formulation, and then preclinical, then in clinical studies. He has did lot of effort to for drug development and the process. So for that purpose, he has paid that much of amount. So that is the reason that price is high to the innovative. But here, after the innovative product is patent is going to be expired, so another companies are formulating like ANDA formulate the same compound, the same formula, and just they will do the bioavailability and bioequivalency studies, BAB. They will not go for the lead optimization studies, identification, and preclinical. Even also clinical also not going to the patient studies. Only healthy human volunteer studies like phase 3 studies, they will go with the healthy human volunteer studies, and they will check the bioavailability and bioequivalency of that compounds. That means that is nothing but the comparison studies, BABE studies. So for that comparison studies, we need the equipment, such type of. So this is type of another type of drug discovery and process. So in everything, we have to go for the GX. That means good laboratory practice or good documentation practice or good manufacturing practice. Nothing but regulatory. So whatever you are doing this work, it should be answerable to the regulatory. So then only it will filing to the regulatory, and it will well known acceptable. Otherwise, they will not acceptable with the good documentation practice or good laboratory practice. So coming to the drug process of development process, HRMS will do all this role, like in drug discovery process, and as in early development and late development. In the early development and late development, the force degradation studies and process impurities. Even once you have to do the force degradation studies, you have to so many possibilities of impurities, possibilities more. So you have to identify all the impurities of force degradation studies. After that, then in the process impurities, whether the process impurities leading to cause any DNA sequencing and all, so that is called as a genotoxic impurities. Even the small quantities also, it is creating the gene modifications, so that leads in different diseases. So that is called as a genotoxic impurities. So it's very low concentration. So that is the reason we have to identify using of sophisticated LCMS technology like HRMS technology, or maybe high-end Tim quadrupole technology for the quantification, and then long-term stability studies and quality control studies. So coming to the another orthogonal technique, just I have explained. If the compound having like peptide, if the peptide molecule having same structure, same parent mass, same fragment mass, even parent, how many fragments are there? All fragments are in the same mass. So then how can able to identify by using, even though it is giving the exact mass also, that all fragments giving with exact mass, then we cannot identify. Like isobaric compounds. Isobaric compounds is the like just only the shape is different. For example, the in layman language, just I will explain this IMS technology is nothing but just you have to take a A4 white paper sheet. So you have to cut in 2 pieces, exact pieces. The 2 pieces are theoretically same. So like same size, same width and everything. But after that, you have to one paper, you have to crush it, and same another paper, you have to keep remain same. So then you have to throw from certain distance. so like 2 feet or from 3 feet above, you have to just leave two papers at a time. One is like a wrap paper, and another one is like one is folded paper, and another is there is like straight paper. When you have to throw down, then first the what is that compressed paper, that the folded paper will come faster compared to the plain paper. Why? Because we have there in we have transferred the material through the air, so the air interruption will there to travel down to the earth. So the harder shape, that is compressed shape, is easily reached to the time taking to reach the folded one is compared to the non-folded one. So in the such a way, we have to identify by using of shape. That IMS technology will identify the compounds by using of another orthogonal technique as a shape. So how it will separate? So we have before the quadrupole, we have introduced one electrical field, the drift tube. The drift tube having the it contains the gas phase in that. So in that ions there are separated that mass by shape and size. So here the larger molecular the traveling toward the gases, the higher the larger is heavy weight or lighter weight. So the lighter weight is moving, and the higher weight, the heavier weight are traveling. So in that cases, it is reaching to the, it is separating whether that depends upon the shape. So benefits, what are the benefits of this orthogonal technique? So in the 3-dimension techniques like AICTE means like retention time, this LC based identification, CCS means collision cross section. So just I explained that IMS the size and shape is giving as the collision cross section. So collision cross section is the one particular value, like how the compound having physicochemical properties, like every compound has having the pKa. So pKa value, pH value. So like that, every compound having one CCS value. The CCS value is unique to that particular compound. So based upon the CCS value, we have to identify the exact compound of that. So then only it is going to the exact compounds. Like in lead optimization, we are almost 5,000 compounds. In the preclinical stage, it is coming to the 500 compounds. Then clinical trials, it is giving the 5 compounds. The final, it is coming output is only 1. So in the only one, if having any other impurity, it is not like there is no efficacy, only toxicity is there. You have to identify. So in that reason, you have to identify the exact lead compound by using of CCS. There is no false positives occur. So coming to the CCS value, what is the importance of CCS value? This is the research publication made. So American Society of Mass Spectrometry is they have published what is the importance of this ion mobility and how it will differ and how it identify by using the large molecular and protein peptide identification. This is the publication. So worldwide, it well-known, identified and accepted methods. So this is the coming to the in market. We are offering different portfolios of different type of instrument, depends upon the customer budget and application we are positioning. So type of application, based upon application, we will position this different type of HRMS instruments, or either it may be single quad, twin quad, or HRMS. So with this, I have completed my presentation. So thank you very much. Given this wonderful opportunity to present over here. Yes, sir.
Thank you, sir, for your nice presentation. You have designed your presentation, which is very essential in our drug discovery and development process. Actually, you have included all parts of emerging technology of LCMS in drug development. So it is very useful for the research scholars who are involved in that area. So we are very much thankful to you, sir.
Thank you, Mr. So, sir, can I leave, sir, or can I wait?
Wait, sir, 1 minute.
Okay.
1 minute. डायरेक्ट कॉल भी नहीं मारता हाँ तुम तो ऑफ कर दे म्यूट कर दे बस अलग कर a positive response in your presentation.
Thank you, sir.
Yes.
Thank you, Bhupinder.
We are sorry for network issues, sir. Somewhat we are facing
Okay.
problem. So I request our principal, sir, to felicitate you with your e-certificate as a token of love and affection. So please, sir.
Thank you, Mr. Ramesh, for your valuable information about this
Thank you.
LCMS, and thank you also for accepting our invitation and coming forward to share your knowledge with all of us. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present you the e-certificate of appreciation. Please accept it.
Sure, sir. Thank you, sir.
Thank you, Ramesh, sir.
Thank you,
Thank
sir,
you.
once again. Thank you, sir.
Bye, sir.
Thank you.